All right, and so today on this day 3 of the fNIRS Bootcamp, we're going to finish up by talking about some of the analyses you can do with your fNIRS data once you've collected it and preprocessed it. And then there'll be some slides on, now that you've analyzed everything, you have some really cool results, how do you share them with people? What's the best way to illustrate and report your results? So it would be steps 4 and 5 of the remaining operating pipeline. Now once you've preprocessed your files, you're typically going to be left with a matrix of your deoxy and oxygenated hemoglobin signal. This will be in the form of time points in the rows and channels along the columns. And from here you want to decide what kind of statistical processes you're going to use to answer your questions about the brain and mind. So recall that in the day 1 we talked about some study designs that you can think about when you're starting fNIRS research. And these come in like different families like general linear modeling, time dynamics, brain state changes. And each of these have different approaches to thinking about data. And that continues in the way that you think about analyzing your data as well. So for analyzing general linear model type studies in fNIRS, it's done similarly to fMRI because our signal is essentially the same. Though there aren't as many fMRI specific steps like scalp extraction or morphing the brains into a common MRI space. What you would do after preprocessing and computing the hemodynamic states, you specify the first level models. And that means, choosing your hemodynamic response function, or if you want to do finite impulse response function models, you'd then enter in your event files to create your design matrix, etc. And then that would create a first level model for each subject. That's essentially generating the betas for how well does this model fit your subject's data. And then you add those into a second level model where you are essentially saying, okay, across all of these first level models, how well does it perform generally? You can also enter any between subjects effects at this point, or look at the consistency effects across people. There are several software packages available for doing GLM with your fNIRS data, though at this time I think they have some large holes in flexibility or usability, especially compared to the stuff that we're used to with fMRI analysis like SPM and AFNI. The ones that I've played with the most are NIRS Lab, NIRS SPM, NIRS Analyzer, and then the NIRS addition to Brainstorm, which is usually used for like EEG and MEG analysis. So far, I like the NIRS Analyzer the best for general linear modeling because it's still in active development. It has some functional connectivity support, and the authors of Ted Hubbert's lab are active and communicative in the fNIRS community. So where there are things that are kind of confusing in the documentation, or if we find bugs, they're pretty responsive to questions and comments about that. But the biggest drawbacks to these packages right now are that most can't do anything more complex than kind of a normal condition comparison. So like no parametric modeling or adding behavioral regressors. And if they do support it, it's not easy to do because few of these have a GUI that it is fully extended for all the capability of the program. So you would have to build up a script pipeline in MATLAB or something instead of being able to click buttons. And then many are MATLAB based and dependent on old MATLAB versions. So if you are not a fan of using MATLAB, that would be limiting. And then if you only have like the newer versions of MATLAB, which is what like hypothetically you should be doing to keep up with code developments, some of these won't work for those newer MATLAB versions because they haven't kept up with the developments and they don't have compatibility with new versions of MATLAB. But besides general linear modeling, there are some other more fun analyses you can do. And at this point, you pretty much are going to be depending on some custom code. There are a couple packages out there that are available publicly that do things more complicated than general linear modeling. For instance, there's an fNIRS that does a functional connectivity thing, and it's a tool. It's like just a specific library instead of a fully completed analysis software. And then the NIRS analyzer has some synchrony functions, but mostly doing this kind of analysis will require your ability to use and tweak custom code. It's more complex than just comparing point estimates of people, which is why a lot of software doesn't support it yet. You essentially are finding ways to compare long data traces instead of just single points of data. So since there's not a ton of previously established software for doing these more complex analyses, I'll give you more of a tour through the kinds of analyses that I've been doing and I've been writing code for, so that way you can understand what these are like, see if it's something that you might want to do, and then there will be some practice we can do with the code that I've already developed. So in the kind of step up from point estimation, that's kind of a one-dimensional data analysis way of looking at it. Neural synchrony is I guess the point estimate would be 0 dimensions and synchrony would be 1 dimensions. Essentially, instead of relying on the amount of activity happening over a time average window, you are therefore caring about amounts, not caring about time. This looks at fluctuations in brain activity through time. And so in this instance, you're caring about variations, not really about the amount of activity at any certain point. This is most often done using Pearsons correlation between time courses across people in the same brain areas. But there are variations on this approach. For instance, you can look at the correlations in time courses between different areas of the brain between people. And you can look at really any combination of areas instead of just one-to-one mappings between people. This is good for questions like, are speech generation processes being instantiated in language comprehension areas in a listener? There are some difficulties with this, that there are many more comparisons to consider if you're doing this any pairwise neural synchrony instead of 1 ROI to 1 ROI. For instance, on our full head setup, we would do, if we're doing a ROI to ROI correlation, since we have 108 channels, that would be 108 comparisons to look through. If we were doing any ROI to any ROI, that would be looking at 108 pairs of channels for each of the 108 channels, and that means there'd be something like almost 6,000 correlations to consider, and so that becomes a very expansive and potentially troublesome multiple comparison problem. Other things that you can do are considering, instead of the synchrony of 2 time traces over the course of an entire scan, like maybe an entire social interaction, an entire movie, you can look at average synchrony over a narrower window of time, and then see how, if you make that window shift through the entire run, then you can see how synchrony changed over time. You can also get more instantaneous measures of synchrony this way with other math that's not just Pearsons correlations. You can ask questions about lagged synchrony, such as if you took 2 data traces and then kind of shifted them in time, which lag amount is going to make the best coherence between people. But still though, since we're looking at many windows of time, many lag shifts, this is going to be another instance where you have to think about your multiple comparisons to keep an eye on. And then besides Pearson's correlations, you can also use other statistical measures such as a robust correlation. And this is essentially just correlation, but the contribution to correlation coming from the autocorrelation of a signal is downweighted. So because our brain signals are autocorrelated, meaning some value at time point t plus 1 is very similar to some time point at t before that t plus 1. So t0 and then t plus 1 will be similar values. And so if you have 2 data traces between 2 people's brains, it could just be the case that this autocorrelation means they happen to overlap in the fluctuations of each other without any meaningful amount of interaction or shared neural processing going on between them. And so robust correlation is just downweighting that autocorrelation to see, okay, besides the contribution to t plus 1 that t0 is giving, are the changes from time point to time point correlated, not just, the data trace itself. Some other statistical measures you can use are called phase synchronization. This is building on the fact that you can decompose a signal into both its amplitude and its position in a oscillation. So because our signal is non-stationary, it's going up and down in a kind of regular oscillatory pattern. You can harken back to 8th grade geometry and remember that in these sort of cyclical patterns, you could plot the data on sort of a circle instead of just an XY plane. So you would use math called the Hilbert transform to do this, to separate off a signal into its phase and its amplitude. And then you can look at whether two people's brain time courses are at the same position in phase instead of just is their kind of data trace correlated. And this gives you an instantaneous measure of synchrony. And this will be more familiar as a process to people who do EEG filtering, because this is something that's pretty common there. And because you're getting instantaneous synchrony, you're preserving more power in your ability to look at across time. If you wanted to look at a kind of moving window correlation on your time series, you can either pick a big window, which will give you lots of data points in that correlation to consider, so your correlation will be stable, but then you can't look at many different windows to see how things are changing over time. In contrast, if you use a small window, you can look at many different windows, but the amount of data points in each window is going to be smaller, so your correlation estimate is going to be unstable. Phase synchronization gets past all that, so every single data point you recorded is going to have some phase value. And there's also something called the Wavelet transform coherence. And this is kind of complicated, but you could understand it as sort of how well does this mother wavelet function, kind of a particular wave shape, match both people's data. And this will give you a across time moment-to-moment estimation of synchronization. It'll give you estimations of synchronization across different frequencies in your data. So in the sort of long fluctuation time course or in the shorter fluctuations that are going on. And it'll also tell you the amount of lag happening. So on this plot here, this is a output of a wavelet transform coherence analysis. And you can see along the bottom that's time from 0 to something like 4,000 time points in this scan they did. And along the y-axis is period, so that's just the particular frequency bin of the signal they're looking into. And you'll see these blobs of red around period 64 that are kind of coming in and out through the time series. That is actually in this study about the frequency period of each trial they were having these people do. They had to, I think, try to mimic each other's button pressing at the same time, and so the prompt for pushing the button would come on at about this frequency. And so you can see periods where they were particularly good at synchronizing that and not. And the arrows indicate which data trace is kind of leading the other a little bit. Now for all of these time dynamics measures, you're going to be wanting to think about your null that you are comparing your experimental estimation to. In GLM, it's easier to think about the null, so you can build stimuli blocks of a condition that contrasts with the one you're interested in, or you can compare your conditions to the rest in between blocks. But in synchrony studies, it may not be good to assume that 0 is the null value to compare your recordings against. And this can be because like we talked about autocorrelation being a potential issue, there might just be the case that there's some residual correlation going on that's not the result of anything meaningful psychologically. It's just not going to be exactly zero. And then as well, our values are not normally distributed, as you can see on this histogram here on the right. So a good idea with synchrony studies is to think about how you're going to bootstrap a null condition instead of just assuming zero is your null. And bootstrapping means shuffling participant memberships in groups. If you are looking at group-based differences, this could be recording a scan of rest, where someone's just sitting there not doing anything, and so you can compare correlations in that rest to correlations to when they're actually watching the stimuli you care about, things like that. You want to create an experimental null distribution that you then use to compare to the distribution of active data that you care about. And then you can consider data that's even more complex than time courses, where time courses look at brain activity values in one place in space across time. You can consider data that's across time and across space. This would be getting into the state dynamics sort of analyses. If you think about brain states, you know, if someone says, I'm in a current brain state, you think like the brain is doing this particular thing in a coherent manner. But across all our measurements, that means all voxels are in like a particular position that is characteristic of this state. So in our data analyses, if we're trying to do state analyses, we can look at this brain state by taking a picture of the brain at one time point and using all the voxels of that brain, or in fNIRS, all the channels, and treating all those channels as one vector. Then take another picture of the brain at time 1, and you have another vector of your all your channels, all your voxels in your brain. If you keep doing that, you can build up what's called a similarity matrix, which is just the correlation of one brain state, that vector of all channels, correlated with the brain state at another time point. And it turns out that the brain is not constantly moving through these states. It sort of sits in a brain state for a while, it's pretty stable, and then it'll suddenly shift to another brain state. And so when you look at these similarity matrices, you can see kind of chunks of time when the brain state is being consistent and then when it's shifting. If you thought about this more vaguely, if each of these voxels is a coordinate in a high-dimensional brain state space, then event boundaries would correspond to when the brain moves through the space, like this. Now in dyads, two people might not be in the same brain state, i.e. having the same brain activation patterns, so maybe their neural synchrony analyses would not be very meaningful. But if they're sharing the experience, they could have mental event boundaries at the same time. So perhaps the more in tune with one another they are, the better their brain state coherence is. You know, think about a dance. There's kinds of dances where everyone is doing the same moves together, and then there's kinds of dances where people have different choreography, but it's very clearly choreographed to the same music. They're all coordinated with each other. So you can think about brain states as these discrete categorical things that the brain is passing in and out of. Prior research that has done it this way uses a hidden Markov model to do this. It's where you're detecting cohesive changes in voxel sets that are happening, assuming some latent event structure underneath. You can find which kind of event structure this would be, you know, number of discrete events, by training it on a data set and then fitting that to a held out subject's data. Or you could ask how well a brain region's activity fits a known event structure, such as the scenes in a movie. Or how well do people's data fit these known event boundaries? Are some people better at shifting the events than others? For this process to work, you either need to assume a known event structure or assume that consistency between people is going to make this work. So if you don't know the event structure, then you just have to assume that people would have the same event structure so that you can then estimate what that event structure would be. In fMRI, you can do this on an ROI to ROI basis, so you can look at different brain areas and how their events, or how their kind of activity moves through events. In fNIRS, where we have many fewer channels, you probably want to do this as a whole brain sort of analysis than just ROIs. You can also treat events boundaries as fuzzy instead of discrete. So you're not looking for discrete boundaries, but you'd be looking for the correlation value between state at T1 and state at T2, just shifting in how correlated that is. So take an off-diagonal from one person's state similarity matrix. This would be their state transition vector. The values of off-diagonal k, whatever off-diagonal you took, tells us how brain state at T1 correlates with brain state at T1 plus k. For this to work, you either need to assume a known off-diagonal, you're not looking at sort of numbers of discrete events at this point, but you'd still be wanting to consider how long do I think brain states are roughly going to be lasting for? Or you would need to, again, assume some sort of timing consistency between people to make this work. For instance, that like a 15-second off-diagonal is where average pairwise correlations between state transition vectors is maximized. No matter which way you do it, the sort of discrete treatment or continuous treatment, you can look at brain state dynamics over time. You can look at changes in time. You can do a sort of moving window version of this to see how easy it is to detect states, say at the first 4 minutes of a social interaction versus the last 4 minutes. And so you can essentially treat your state dynamics data like you would think about your neural synchrony data. You're just on a sort of two-dimensional representation of your data instead of one-dimensional. And other kinds of state dynamics questions you can ask, like what's the inertia of the system? Inertia in the dynamical systems sense means if you're in a state, how hard is it to shift out of that state into a different state? You can ask about lags again. You can ask about state dissipation. If you are in certain states, does the amount of time you're spending in states stay consistent through whatever this experience is, or do you start like shifting states more quickly or more slowly? You can ask about recurrence. The way we were looking at brain states in the previous plots was that we're just looking at when are these brain transitions happening, but we're not asking are any particular brain states reoccurring at some other time point. You could ask that question, which would be more rigorous of a question to ask of your data, but it could be very interesting. I put here some links to good dynamical systems work in behavioral data that has really inspired the way I think about state dynamics and brain data. You can look at this Richardson, Dale, and March chapter that's linked there. Butler and Randall 2013 looks at dynamics between dyads in close relationships. And they give you lots of interesting ways to think about your data that you might have thought of before. Also, since these are two things, neural synchrony and state dynamics, that I've played around with before, I'm going to post the code that I've written so far for doing these to the GitHub that we can then play around with and practice. But beyond that, there are still other ways of looking at data that are really interesting and computational and really leverage the fact that you're collecting lots of data across lots of spaces or places in the brain and in interesting situations between people. For instance, you can do network studies. What's the network shape between people? Here at UCLA, Carolyn Parkinson does some interesting work where she looks at kind of how either people's brain activity kind of looked at normally is characteristic of their position in a social network, or how other people look at it. You can look at a single brain as the social network itself, different regions of interest being the different nodes in the network, and seeing how different regions correspond to others and what's the functional connectivity going on. you could expand that into considering what's the functional connectivity between nodes in other people's brains. What's the shape of this network that's emerging? How dense or efficiently is it structured? For doing these kinds of analyses between people, you would use what's called multilayer networks. And essentially, it's a version of multi-level modeling, but for networks. It takes into account the different networks that may be connected to each other. You would use this for analyzing networks between people, networks over time points, etc. Some good papers to read are definitely from Dani Bassett's work and her collaborators, so I posted some papers there that I really like. You can also do something called kind of dimensionality studies. I would say this probably has the strongest like machine learning flavor of the stuff that I'm presenting. Even though in a very most technical sense, all of these are machine learning because, you know, a regression is machine learning. But if you're thinking about it in a more vague sense of making decisions from complex data that's dividing it into dimensions that I didn't tell it to or don't understand, and that it's just cool and sounds impressive to people, this would probably be the closest to that conceptualization. This is essentially making principal component analysis your phenomenon of interest rather than just a method to clean data. So across time and space, how much information is represented in your brain data? How many distinct dimensions could you reduce it to without losing important variation? How complex or simple is your data? You would use algorithms such as principal component analysis, k-means clustering, mutual information, etc. And fNIRS, using this approach, you might ask, is the dimensionality doubled between 1 and 2 people doing a task? Or is there some shared information between them? What tasks would reduce the dimensionality between people, meaning they are sharing more brain activity between each other? And so that's kind of a high-level overview of potential approaches you can use to analyzing your fNIRS data. This is definitely not all the things you could ever do with analyzing data. Of course, there's going to be many, many more things yet to be discovered, or many more things that I just didn't talk about, but other people are doing that are really cool and that I don't understand. This is not a finite set of ways to analyze your data, just some interesting things that I've come across so far that I think are fun and might be good for our lab to think about.